Good morning to everyone. Today we will discuss about to the cell division. The cell division to the two types. One is the mitosis and meiosis. Now we will discuss about to the mitosis. What is the mitosis? The cell division mitosis is an integral part of the cell cycle. The production of genetically identical daughter cell division is called the mitosis. Here cell duplicates their genetic materials before they divide, ensuring that each daughter cell receive an exact copy of the genetic material. The genetic material is called the DNA. Here after cell division to divided into two daughter cell. You can observe to the mitosis, this is the starting process. Each and every cell to divide into the form of the two daughter cells. Here what is the DNA? First discuss about to the genetic information. DNA is the genetic information. You can observe the in the nucleus genetic material is present, that is the nucleic acid. What are the nucleic acid? Nucleic acid two types, one is the DNA and second one is the RNA. DNA is the main important of the genetic information one generation to another generation depending the time. Sometimes these genetic material found into the chromosomes also, depending the time, depending the division, in that time the DNA, chromosomes all are the carry forwarding to the genetic information that one generation to another generation. This is called the genome or gene. Here the DNA, genetic material, the genome and chromosomes all are the family of the genes. Here what type of chromosomes are present? Packaged chromosomes. This is called the packaged chromosomes and inside the nucleus to present. What are the chromosome duplication? Chromosome structure in the centromere is there, two chromatids are there, this is a common chromosome. Because of the duplication, each considered chromosome consists of two identical chromatids. The structure of chromosome you can observe in the middle centromere is there and two arms are there, that is called the chromatids. This is the structure of chromosome. You can observe the structure of clear chromosome only where in anaphase, telophase, like that only. But where the prophase is there, that is a thread like structures are present. Here the chromosomes two identical centromere and two chromatids are present. What is the duplication? Here because of the duplication, each and every chromosome condensed consists of two identical chromatids joined by a centromere. Each duplicated chromosomes contain two identical DNA molecules. These identical DNA molecules meaning unless a mutation occurred, that is called the two identical DNA molecules, one in each chromatid. This is the chromosomal duplication. Coming to the our topic mitosis. What is the mitosis? Where you can observe and what purpose, what type of significance or importance to mitosis? Mitosis term derived from Greek word mitos. The meaning of mitos meaning threads or fibrils. This is the meaning of mitos. Where you can observe the mitosis, parent cell produced two daughter cells, that is called the mitosis. The parent cell after division of the mitosis to form to the two daughter cells. Here these two daughter cells, identical daughter cells that are genetically identical to the parent cells. This is the mitosis. The meaning of mitosis, all mitosis chromosomes, thread like structures are there. That is the meaning of the mitosis. Okay, the first observed the mitosis Strasburger scientist. You have studied to the in the plants only, no, the mitosis you can observe animals and plants, but here particularly we will discuss about to the plant cell. In the plant cell, the mitosis to observe the Strasburger scientist in 1870. Another, the second scientist Boveri and Fleming scientist to observe the animal cells. The term mitosis to given by the scientist Fleming scientist in 1882. What is the mitosis? The mitosis called also equational division due to equational distribution of the chromosome in a daughter nuclei, also called stomatic cell division, where you can observe the mitosis. Okay, many cells are present, but where the mitosis you can observe, where the somatic cells are there, in that somatic cell division mitosis under present. Okay, what is the somatic cell? Here all body cells except gamete cell, gamete and male and female gamete, where you can take the animals or plants, male gamete is there, female gamete is there, except the gamete cells where the all cells of the stomatic cell under mitosis is present. This is a mitosis process. In the mitosis, the site of mitosis in plant is observed to the meristematic region, where you can observe the divisions are there, meristematic division, the plant how to growth after division, what type of tissues are important in the division, that is the meristematic tissue is the most important. In the meristematic tissue, where you can observe root apex, shoot apex, intercalary meristems, lateral meristems, leaves, embryos and seeds. These are the basic divisions of the plants. In the root apex, you know, root apex epiderm is there, all present of the peribulum, these are the present. In the root apex calyptrogen is there, in that calyptrogen the divisions are more, that division name is called the mitosis. Coming to the shoot apex, here shoot meaning stem. How to stem to growth? In the stem also many scientists to explain the stem theories also, histogen theory, corpus kappa theory, like that. In the shoot apex also the tip of shoot, many meristematic tissues are there, in that tissues or in that cells two mitosis divisions are present. Coming to the lateral, in the meristematic tissues the lateral is there, intercalary meristems are there, where the intercalary, where the lateral meristems are there, in that meristems also under the mitosis process is present. Coming to the leaves, you know how to leaves are there, in the leaves also the meristematic, under meristematic tissue the divisions are present, that is called the mitosis division. Coming to the embryos, same, embryo also the division is the mitosis, where the development of seeds also to mitosis after mitotic division, the important the develop the seeds also. These are the plant body you can observe the mitosis. Coming to the animals, in animals embryo, skin, bone marrows are the mitotic divisions place in the animal. These places are the most important for the animal tissue. Embryo is one, the skin, where the skin is there, in that skin cell where the bone marrow is there, in that bone marrow the under the mitosis divisions are present. Okay, in the plant body what is the mitosis, how to divide? Some haploid and diploid cells, you know both haploid is the single, diploid is the double. These haploid and diploid cells divided by a mitosis. Each and every new cells receive one copy of every chromosome that was present in the original cell. This is produced two new cells that are the both genetically identically to the original cell. This is the mitosis, haploid and diploid. Coming to the diploid cell, after first diploid cell undergo DNA duplication during the interphase to form to the separation of the chromosome. After mitosis, you can observe to the two cells, the parent cell after mitotic division to form to the two daughter cells. In the mitosis, okay, the cell is division is there. In the cell, different cell organelles are present. You can observe to the in the cell cytoplasm is present. In the cell many cell organelles also present. Many cell organelles, the most important brain of cell is called nucleus. In that nucleus, the DNA, RNA, chromosomes or genes, genetic material found inside the nucleus only. This one here, the division of the mitosis, two types. One is the karyokinesis, The second one is the cytokinesis. What is the karyokinesis? Where the nucleus is there, in that nucleus the DNA, the nuclear divisions are there. Nucleus division is called, or nucleoplasm division is called karyokinesis. Where the cytoplasm, in the cell the fluid structures are there, fluid like formation is present, that is called cytoplasm. That cytoplasm divisions are there, that is called the cytokinesis division. This is the mitosis basic phases, 2 phases total the life cycle of the mitosis. Here in both karyokinesis and cytokinesis, both mitotic divisions are present. First you can take the karyokinesis, nuclear division of parent cell known as karyokinesis. What is the meaning of karyokinesis? This is a Greek word, karyon meaning nucleus, kinesis meaning movement. These karyokinesis again divided into the 4 phases, that is called prophase, metaphase, anaphase and telophase. These 4 phases observed in the karyokinesis. What is the prophase? This is a mitosis, the first division of the karyokinesis, first division prophase. Here you can observe the prophase can be divided into again 2 parts, early prophase, mid prophase, late prophase. Here early prophase is the all chromosomes thread like, and mid prophase is the like these chromosomes are some bulge areas are present. Late prophase you can observe to the division very clearly. Early prophase, what is the early prophase? DNA molecules condensed and form a short chromosome. First observe the prophase, the chromosome size is decreased, that is why condensed or short in size. In this stage, chromosomes appear like a ball of wool, so called, that is the spime stage or thread like stage are there. And in case animal cells, the centrosome, that is the centriole, divided in 2 and move towards opposite poles. This is the early prophase. What are the spindles operators? Here the chromosomes are also called achromatic structure. This is the centrioles, ester, spindle fibers, all are the mitotic spindle operators. In here the operators of microtubules control the chromosome movement during the mitosis time. Here the centrosome replicate to forming the two centrosomes and that migrate to the opposite ends of the cell. This is the spindle microtubules begin in the centrosome, the microtubules organizing centrole, a asters. This is a radial array of short microtubules extend from each chromosomes. This is the spindle operators from the chromosomes. Coming to the mid prophase. What is the mid? Chromosome shift towards the periphery and leave a clear area, that is the mid prophase. It become short and thick. Each and every chromosome consists of 2 longitudinal threads, that is called the chromatid. That name is called chromatid. Coming to the last one, late prophase. In the late prophase, spindle fibers start appearing around the nucleus. The size of chromosome is so much reduced. The spindle folds are formed without a asters in plant and with asters in animals. This is the late prophase. The second phase of karyokinesis, metaphase. Metaphase is the longest stage of the mitosis. At least about 20 minutes time to take the metaphase. Here the centrosome are now at opposite ends of the cell. The chromosome arranged on the metaphase plate. This metaphase plate meaning equatorial plate. All chromosome comes to the middle part. That middle part is called the equatorial plate. This equatorial plate where the Golgi complex is there, in that Golgi complex to produce the equatorial plate. Here all chromosomes is the congressive, or all chromosome you can observe the middle plate. Here the chromosomes centromeres lie on the metaphasic plate. For each chromosome, the kinetochore of the sister chromatids are attached to the kinetochore microtubules coming from opposite poles. The entire apparatus of microtubules is called spindles because of its shape. This phase is useful for study of chromosome morphology. You can observe the chromosome. Prophase you can't see chromosomes, only thread like structures are there. You can't observe to the centriole or chromatid. You can observe the chromosome structure only metaphase. In that phase the centromere is important, and the two chromatids also very important in the metaphase. Coming to the third phase, anaphase. Anaphase is very shortest phase. Here centromeres chromosomes start divided into two daughter chromatids are separated. Here the two daughter chromatids are migrate towards the opposite poles. Here the spindle fibers are attached to the centromere and pull the chromosome to the opposite poles. This is the anaphase. Coming to the anaphase, the kinetochores microtubules is very short in size. Because these microtubules are attached at the centromere region, the chromosomes move centromere first. The cell elongation as the non-kinetochore microtubules length. By the end of anaphase, two groups of chromosomes formed, one at each pole. Anaphase you can observe the different shapes of chromosome. This is the shapes of chromosome. In the study of the chromosomes time, the structure depending the anaphase time, the chromosome you can observe the prophase time, the chromosome only thread like. Where the metaphase is there, you can observe the chromosome structure very clear. The centromere is there, two chromatids are present. But where the anaphase is there, in that anaphase you can observe the chromosome different shape, V, L, J, I shape. V shape is the median shape, that is the metacentric shape. L shape sub-metacentric sub-medium shape. J shape acrocentric shape, that is a sub-terminal shape. The last one is the I shape, that is a telocentric or terminal shape. Last coming to the telophase. Here in the telophase, this is a cytoplasm discretions. Nuclear envelope is formed around each set of chromosomes. Chromosomes from chromatin nucleolus is reformed. The spindle fibers or spindle apparatus here disappear. The mitosis, the cell division, one nucleus into two genetically identical nucleus is now complete. This phase is just reverse of the prophase. You can observe the prophase structure, and this telophase just reverse of the prophase. This is the telophase. In plant cells, the mitosis in plant cell, this is the prophase structure, and prophase the chromosomes condensing, the nucleolus is beginning to disappear, although not yet visible in the micrographic. The mitotic spindles are starting to form. This is the prophase. And prometaphase and metaphase. We now see describe the chromosome. Each consists of two identical sisters chromatids later in a presence of the late phase of the metaphase. Coming to the metaphase, the spindle fibers complete and chromosomes attached to the microtubules, their kinetochores, and this one is the metaphasic structure. Anaphase, the chromosomes, each of chromosomes have a separate and daughter chromatid are moving to the ends of the cell. Their kinetochore microtubules are very short. Coming to the last one, telophase. Daughter nuclei are forming. The meanwhile, cytokinesis has started the cell fate. With wall divided, the cytoplasm divisions are there, and to forming two 2 is growing towards the pole side or parental side. Coming to the, this is the animal cell of the cytokinesis. Here 2 havels are there. Middle region of cell, microfilaments induce the cell membrane, and the ferro formation depend centripetally and finally cleave the parent cell into 2 daughter cells. This method is called cleavage method. You can observe only animals, and sometimes plant cells also. See, plant cells and difference from the animal cell. This is a cleavage, ferros are present in animal cell. The plant cell, vesicles forming cell plate to form very clearly. This is a cell plate formation in plant cell, that is the stem. Where the animal cell is there, that is called the cleavage of the animal cell division. What are the significance? Okay, after mitosis, the significance of the mitosis, genetic stability is present. Here, mitosis produced 2 nuclei which have the same number of chromosomes as the parent cell. Daughter cells are genetically identically to the parent cell, and no variation in genetic information can be introduced during the mitosis. This results in genetic stability within population of cell derived from the same parental cell. These are the significance mitosis. In the significance, what is growth, what is cell replication, what is the regeneration, what is the asexual reproduction. First one, growth. The number of cell within organism increases by mitosis, and this is the basis of the growth of multicellular organisms. This is a growth. And what is the cell replacement? Replacement of the cell and tissues involved in mitosis. What is the regeneration? Some animals are able to regenerate whole parts of the body, such as legs in crustacea, arms in starfish. Production of the new cell involved in the mitosis. What is the asexual reproduction? Mitosis is the basis of the asexual reproduction. Their production of new individuals of species by one parent organism, that is called the asexual reproduction. This asexual reproduction is complete to the mitotic cell. In the mitosis, you can observe to the plants, animals is the most important. In that one, you can observe to the mitosis prophase 1, prophase 2 and metaphase, anaphase, telophase. These are the completion of the mitosis. In the next, coming to the mitosis, here cell divisions also there, that is the gap 1 phase, gap 2 phase and gap 3 phase. Where the gap 1 phase is there, in that gap 1 phase you can observe the starting divisions are present in the mitosis time. This is the gap 1 phase. Coming to the gap 2 phase, gap 1 is the lengthy time and gap 2 is the another lengthy time is present. In between, S synthesis are there. S synthesis meaning here DNA replications are present, DNA synthesis are present. Here this phase total, this is called the between the interphase of the mitosis. In the interphase, you can observe the before mitosis, interphase is the most important all cell organelles, that is the plant cell organelles or animal cell organelles. That one is the interphase. In that, after interphase, in the plants you can observe to the this prophase 1, metaphase 1 and second one is the anaphase, telophase and last one is the telophase. Here the prophase 1 you can observe to the chromosomes only, just like thread like structures are present. You can't see observe to the chromosomes, it's not clearly. What is the structure? In the chromosome you can observe the structure, centromere is there and 2 arms are present, that is the chromatids are present. These are the importance of the chromosomes. You can't see particular prophase. Coming to the anaphase. Anaphase, you can observe to the anaphase. Here chromosomes are the just increase in size. Increase in size meaning the centromere is particularly you can observe the clear, And coming to the chromatids also, very particular points are present. And in this phase, chromosomes all are observed in the different shapes, different structures also. You can observe in the metaphase another important characters, all chromosome condense together in the formation of the cell plate. Here cell plate, many enzymes or many cell organelles are importance are there. Best example, Golgi complex. These Golgi complex phragmoplast are to produce the phragmoplast, in that phragmoplast to form to the equatorial plate. In that equatorial plate, all chromosome combine with the equatorial plate. You can observe the metaphase, in that phase is the observed the all chromosome in equatorial plate, that is called the metaphase. Prophase, you can't observe to the chromosomes condensed and the chromosome size is very less, all are the thread like structure. Where the metaphase you can observe, in the metaphase chromosome size increase and all chromosome you can observe to the in the equatorial plate. Coming to the next one, anaphase. After third phase is the anaphase. This anaphase is the most important for the movement of the chromosome. Each and every where the chromosomes all to attach the centromere, no, in that chromosome move from the pole side. This is called the anaphase. And coming to the after anaphase, here anaphase, the both cell divisions to divide and move from one side to another side. After that one is the telophase. The telophase is the chromosomes you can observe to the move from the end of the end of the cells. The here two daughter nuclei to form all daughter nuclei from the parental nucleus, all characters from the parental nucleus. These are the total mitosis. And you can observe the mitosis only somatic cell in the plant one, and main important here karyokinesis is present, cytokinesis both are present. Karyokinesis is the nucleus. In the nucleus, nuclear envelope is there, nucleoplasm is present, and in the nucleus chromatid like structures, thread like structures are present. That thread like structures name is called chromosome. These chromosomes are main important, not only chromosomes, here DNA is there, chromosomes are there, sometimes utilizing the DNA, sometimes utilizing the chromosome, sometimes utilizing the RNA. These are before the divisions, these are the found in the nucleus. The presence of in the nucleus, these genetic material is found the nucleus cell. In the nucleus, here mitotic division time, the you can observe the chromosome division only. DNA used for the replication. Where the replication you can observe in the cell cycle phase, gap 1, gap 2 phase, in between S synthesis phase is there. In that S synthesis phase, the DNA replications are present. In that time, DNA is utilized. That's why this one is the you can observe the in the cell cycle or cell division time. But here the mitosis time, chromosomes are involvement is there. That's why these chromosomes to divide in the nucleus, the divisions to start in the mitosis. Okay, here the chromosome G, the chromosomes DNA, RNA. RNA is there, no, in that RNA you can observe the only in the protein synthesis time. DNA also different types of DNA, aDNA, B, C and Z DNA are there. RNA also different, mRNA, tRNA, rRNA, messenger RNA, transfer RNA, ribosomal RNA. These RNAs also to all found in this one. Okay, here the total genetic material found in the nucleus only. These genetic material main important of the chromosome to transfer into the karyokinesis into cytoplasm. Here this after karyokinesis total mitotic division to complete and meiotic division to the cytoplasm. The cytoplasm also, here the meaning of the cytoplasm, cytoplasm is inside the cell. The structure of plant cell wall is present, after cell wall plasma membrane is present, a plasma membrane inside plasmolysis, or this one is the gel like structure is there, that is the cytoplasm. This cytoplasm found in many enzymes and many cell organelles are found in. Here in the cytoplasm, the division of cytoplasm is called, that is a cytokinesis division. Both divisions are important for the mitosis, that is the karyokinesis division, and coming to the second is the cytokinesis division. In the cytokinesis, here many cell organelles present, just like the mitochondria, Golgi complex, plastids, vacuoles, nucleus and lysosomes, different types of all cell organelles present. In the cytoplasm, when the division time, the most important cell organelle is the Golgi complex. Here the function of the Golgi complex, the divisions are present in this one Golgi complex to form to the fragments. That fragment to use for the where the mitotic divisions are there. In that mitotic, the second phase, the first one is the prophase, the second one metaphase. In the metaphase, these fragments, the Golgi complex to produce the complex, no, in that migrate to the complex system are formed, that is the phragmoplast. In that phragmoplast, all chromosome to attach the, when the phragmoplast, when attachment is there, that is the use for the Golgi complex. This is used for many times to ask the question, the which in the mitotic stage, which cell organelle to form the cell plate, that is called the Golgi complex. In this one is the total mitotic divisions, and after mitotic divisions you can observe parent to two daughter cells. This is called the mitosis. Thank you.